My name is Michael Levin, and I'm at the Allen Discovery Center at Tufts. Today I'd like to describe a technological approach to mind everywhere, or TAME, a new framework for diverse intelligent research that's based on a continuum of minds in unconventional living agents. The main points of my presentation are the following. How much intelligence or agency or cognition any given system has can be framed as an engineering problem, not just philosophy. I propose an empirical approach using the correct optimal level of cognitive concepts that best predicts and controls any given living system. I provide a framework in which diverse intelligences can be directly compared with respect to the spatiotemporal scale of the goals that they're capable of working towards. This is the cognitive light cone model, and it fundamentally focuses on goal directiveness as a measurable aspect of diverse intelligence. Synthetic bioengineering now provides an incredibly large option space of new bodies and new minds that often don't have standard evolutionary backstories. This provides an incredibly rich pool of novel beings for any diverse intelligence frameworks to consider. And I'm going to talk about the fact that morphogenesis can be viewed as an unconventional collective intelligence, one that solves problems in morphospace, and via our extension of neuroscience methods and mechanisms beyond neurons, being developmental bioelectricity, provides a new window on the problem of how many competent individual subunits are bound into one larger agent with a common centralized intelligence and common goals. So the outline of today looks something like this. We'll talk about the concept of diverse intelligences, and then I will get into the actual approach that we seek to take on this, both in the philosophical principles and the specific framework, and then briefly show you some model systems and data that bear on this, and then I'll describe the actual action plan that we propose and some areas in which we would love to have collaborators in the community. So let's first look at the questions of diverse intelligence. It's already clear now that there are examples of living things that are quite distinct from the standard subjects of intelligence, like higher primates and octopuses and so on. This right here is a physarum. It is a unicellular slime mold. The whole thing is one cell. And you can see here that in this type of problem, it reliably chooses to go towards 3 glass discs instead of 1. And in this model system, what's interesting about it is that its behavior is the same as its morphology. That is, the shape of its body is the behavior that this system exhibits. Now, another important concept in this, starting to think about unconventional substrates, is that the subject of intelligence, that is the agent that we think exhibits some type of diverse intelligence, is in fact quite plastic. So here we have caterpillars, and they have a specific brain with specific behaviors. They need to change into a very different animal that has very different behaviors. And during this process, the brain of the animal is basically liquefied and rebuilt. So the bodies, and in fact the brains of agents, are quite malleable. And it's important to think about not only the diverse intelligence of a sort of finished, completed, standard type of animal, but actually what happens to this intelligence during remodeling and deformation. And we can see further the relationship between anatomy and cognition in something like this. These are planaria. They are highly regenerative, but they're also smart and they can learn. And so planaria that have learned to associate a particular feature with food can be shown to recover that information when their brain, and in fact the whole head, is amputated. So the tail will regenerate a new head, including a new brain, and after that the remaining tissue somehow imprints the original information onto the new brain so that we can observe the behavior in which it once again recognizes that original information. Now, of course, planaria can be cut into many pieces, presumably all of which should have the original information. So it's really interesting to look at the number and the type of agents that can arise during deformations and rearrangements of the pieces that are normally used to construct a standard type of intelligent agent. So in our group, we think about cognition as a nested property, all the way from genetic networks up through subcellular, cellular, tissue level, organ level, and even swarm levels of organization. Because each level of organization solves problems in its own appropriate space. This might be morphospace, or the space of possible anatomies. This might be transcriptional space and figuring out which genes to turn on and off. It might be physiological space. It might be the more commonly thought about the three-dimensional behaviors space, and so on. These are all solved using some of the same mechanisms and very diverse levels of sophistication, from very simple basal intelligences to quite sophisticated ones. But what's really interesting is that modern bioengineering is now providing us with an incredible diversity of agents. So these are not only the typical model systems that have been on Earth and for eons and that we know how to deal with, but these are now hybrid or entirely novel creatures that may be partly biological, partly artificial. They may have pieces of robotics, pieces of software agents and machine learning. All of these bioengineered different types of constructs, various kinds of cyborgs, chimeras, and so on, are really extending this idea of the types of life forms that we need to deal with in diverse intelligence research. And somewhere on this Wiener and Rosenbluth scale here, these agents are all going to have different kinds of cognition, all the way from very simple active or non-active types of behaviors that we can even see in metamaterials and smart materials, all the way up through various feedback-based predictive and other types of more complex cognition. And so we really need frameworks that acknowledge the fact that we can no longer rely on old categories, including how and whether some agent was evolved or designed, or what it looks like, or what kind of components a given animal has, but actually to really embrace the fact that at every scale we might be dealing with completely novel agents, whether at the cellular, individual, or swarm level. You might have combinations of evolved, designed, organic, inorganic, and so on, pieces of highly variable sophistication. Okay, and so we need ways to be able to deal with the types of intelligences that all of these kinds of constructs are going to have. And so we've developed this idea, which is a kind of technological approach to the supposition that there are types of intelligence everywhere, all around us. And so let's talk briefly about the philosophical principles and then some specifics. So what we would like to do is to provide a framework for being able to compare truly diverse intelligences. This is regardless of their composition or origin story. So we're going to not only be able to deal with familiar creatures such as humans, apes, birds, octopuses, and so on, but all kinds of weird biologicals such as colonial organisms, swarms, and swarm intelligences, but also the products of synthetic biology, engineered new life forms, artificial intelligence, whether software or embodied, and eventually potentially exobiological agents. So we want to go beyond the types of biological systems that the Earth's biosphere has provided us, which is really an N of one type of example, and be able to handle and recognize and compare and relate to highly diverse intelligences. And we're really focused on doing this in a very empirical way, so an engineering approach whereby the best theories are going to be ones that provide the most accurate and the most efficient prediction and control of these complex systems. That is how we're going to judge the quality of the models that we have. So the foundations for this are the following. First of all, I propose that having binary categories, such as asking whether something is cognitive or whether it's an agent or whether it makes true decisions or just as if decisions, I think this is false, and I think this is how we get into a lot of trouble by trying to assign these to binary categories where we have some sort of almost magical true cognition and then other things that are just physics but maybe they look like cognition. I think we need to take evolution very seriously. We need to take gradual phylogenetic origins seriously and realize that all of these types of metrics that we're used to, intelligence, cognition, and so on, are gradual and belong on a continuum. And so the real question is how much cognition, memory, intelligence, and so on something has, not whether or not it has it. And the point is not to try to anthropomorphize simple systems, but exactly the opposite, to demystify cognition so that we can say where it is that advanced beings like humans obtained their cognitive capacities and what those looked like all the way back to the most humble origins of life. And so the idea here, having this be an engineering perspective, is that we need to have an empirical stance on these theories, meaning that we can't a priori say that that attributing goals or goal directedness and so on is somehow fundamentally bad. The idea is that type 2 errors, meaning failing to attribute sufficient agency to a system that is actually better predicted and controlled by models with agency, is as bad as assuming excess agency that isn't there, that does not provide any useful prediction or control power. So we're going to try to get away from this teleophobia, where people are really concerned about attributing too much agency, but almost no one worries about insufficient agency, which actually is a major problem holding back regenerative medicine and so on. And so the idea is that we're going to try to pick the correct level of agency that optimally provides prediction and control, not too little and not too much. So this means that in this kind of idea where we attribute cognition to all sorts of unconventional systems, so not just complex organisms with brains, but other types of organisms, and in fact the components of various organisms, we're going to scale it properly. So the point isn't a kind of panpsychism where we think that rocks have hopes and dreams, but in fact correctly scale the types of goals and cognitive processes that things like organs, single cells, and other types of processes may be undergoing. At the same time, we're not going to assume that there's no way to make a mistake. Of course there is. And so, for example, the reason it doesn't help to think about to say that a thermos has to know whether to keep something hot or cold is because there's a perfectly good simpler theory of how it works that does not attribute that level of agency to the thermos. So we're always going to try to attribute just enough agency that provides the optimal prediction and control. Okay, and we're going to assume that agents can be a patchwork of multiple intelligences. So you may have a complex brain and have a single centralized intelligence there, which may even be verbal in some cases, but in fact that body may have additional intelligences in the various physiological and anatomical systems that can be dealt with in a different way. And in particular, we're going to say that there is no magic in the material. That is, intelligence is not a property of synapses, of neurons, of brains, because lots of other types of systems can solve problems and reveal aspects of intelligence, but in fact, not even protoplasm. So we're open to all kinds of substrates as long as they meet appropriate functional goals. And we're really interested in biology, not zoology. So not just specific life forms that we have here on Earth, but really life as it can be and mind as it can be, a full diversity of these things. And the idea is that we are going to define a way to compare highly diverse intelligences by saying that what is central is the ability of various agents to work towards specific goals. That goal directedness is central to a concept of intelligence. It's problem solving towards a preferred outcome, towards a preferred goal. And so one way to compare all of these is through something we call the cognitive light cone. And basically this simply borrows a style of diagram from relativity, where we collapse all of space on one axis, that's the horizontal axis, and then time is on the vertical axis. And we simply plot out the semi-quantitative scale of the types of goals that any given agent can pursue. Not the distance that it can sense, but the scale of the goals that it can work towards. So for example, if there's a tick, the cognitive space is quite small, because basically it only cares about the concentration of specific molecules in its vicinity. As far as we know, it has very little memory backwards, no predictive power forward, so it's got a very small cognitive cone. If you have something like a dog, then it has considerable memory backwards, it has some predictive power forwards, there's a radius of events that it can work to maintain specific states in, but it's never going to care about something that's going to happen two weeks from now in the next town over. It's just simply impossible for that cognition. So you can think about what are the spatiotemporal scales of events that various creatures can work towards. Of course, if you're a human, you might work towards very large, both in terms of future time and in terms of spatial states. You might work towards world peace or something like this. And then, of course, various alien, artificial, and other types of cognitions can have all kinds of different shapes on the scale. And in fact, they can be nested, and they can all be scaled one inside the other. Now, one can see the scaling of the goal space of an agent in this biological example. So what you're seeing here is a lacrimaria. It's one single cell. There are no nerves, no brain, no stem cells, no cell-to-cell communication. And what this system does is handle all of its anatomical, physiological, and behavioral needs all on the scale of one cell. Now, cells like this work together during multicellular metazoan development, and they build something very complex. They work towards a very large-scale goal, building various organs in a body. When deviated from that goal, they know how to undergo novel behaviors to get the pattern back. And so you can see this in the limb regeneration of a salamander. The limb is amputated, and then over time the cells work together as a kind of collective intelligence to rebuild a specific salamander arm shape, and there it is. When it's rebuilt, they stop. Okay, so this is a system that doesn't simply roll forward and produces some sort of emergent outcome, but it actually reacts flexibly to stimuli, and when it's deviated from the correct region of morphospace, meaning having a correctly patterned limb, it is able to undergo novel deformations and traverse that space to get back to where it needs to be, which is, once again, to rebuild that limb. And so now these cells are working together as a kind of group agent on a much larger goal that doesn't belong to any of the individual cells themselves. And this, of course, can break down. The system can break down, and individual cells can defect from this system when they are disconnected, in particularly electrically disconnected from the neighbors, and they basically revert to a unicellular life form. And here you have these are cancer cells that are basically treating the rest of the animal as just environment. You know, the scale of the self has shrunk as far as these cells are concerned, and so that ability to scale the boundary between self and world is really prevalent in biology. And you can see this schematized here, where a single cell has a certain small radius of events that it can manage. It has a little bit of memory going backwards, a little bit of anticipation capacity going forwards in time, but joined together into a network, it actually has it becomes a bigger self. It's got a bigger radius of concern. It has a bigger computational capacity, both for memory and for anticipation. And this type of tracking of the scaling of the self and its goals has very clear predictive capacity in biology, where actually when individual cells begin to electrically decouple from the rest of the collective, and this can happen because of oncogene expression, and various other events, these cells, you can actually see their aberrant electrical potentials. This is a voltage dye map showing where an incipient tumor is going to form. And in fact, this process can be suppressed, and so here the oncogenes, you can see, are very strongly expressed, but there is no tumor, because what we've done is express particular ion channels here that artificially force this cell to remain in electrical connection with their neighbors, to be very tightly informationally coupled, and thus continuing to pursue normal morphogenesis and histogenesis as opposed to going off on its own, and this suppresses tumors. So this is a very practical idea that thinks about the goals that collective or individual agents are going to pursue, that has implications for biomedicine. So let's dig into this a little bit. In particular, this idea that morphogenesis is a collective intelligence that flexibly pursues specific anatomical states. And then I'm going to show you an artificial organism that is an example of this kind of work that I think can be very useful for diverse intelligence research. So where in fact is anatomy specified? You know, we all start life as a collection of embryonic blastomeres, and then what they do is they reliably build this incredibly complex anatomy. So this, of course, is a cross section through a human torso, and you can see all of the individual organs and structures that are in precisely the right size, shape, orientation with each other. And so we know that DNA specifies proteins. Where does the anatomy actually come from? and how does this collective know what to make and, more importantly, when to stop. And for regenerative medicine, we ask questions like, how would we convince a bunch of cells to rebuild one particular structure if it were missing? And then as engineers, we ask, well, how much plasticity is there? Could we get these cells to build something completely different despite their default genome? We've already seen this animal to show that individual cells are extremely competent. And I want to just drill down for a minute on the problem solving and the intelligent plasticity that they can exert when they are collected together and harnessed towards a group goal that is a function of the larger scale individual that is formed. So this is a tadpole. Tadpoles must rearrange their face to become frogs, and the remarkable thing is that all of these organs, if we place them in incorrect positions to begin with, so the eyes are on the back of the head, the jaws are off to the side, the nostrils are in the wrong place, everything's wrong, these animals still remodel to become perfectly normal frogs, because all of these organs will continue to move around in unnatural paths until they get to the correct frog face, and then the whole thing stops. So what the genetics here gives us is not a specifically hardwired set of movements, like each component of the face just moves in the right direction, the right amount, but actually it specifies a very flexible error minimization system that can begin in unexpected states, but still get its job done. It very flexibly pursues this goal of creating a correct frog face. And we know that it's not simply a feed-forward process. It actually is a flexible type of system, because we can make significant perturbations such as this and watch the system expertly navigate that morphospace to get where it needs to be. And so what we've studied in our group are these feedback loops that basically implement a kind of anatomical homeostasis. The idea that the collective agent has set points, and these are not only simple set points that individual cells have, like pH and metabolic state and so on, but actually complex set points that only a collective agent could maintain, represent, and pursue, which are these anatomical layouts. So a complex descriptor that in some coarse-grained way serves as a set point for this homeostasis, so that when the system is perturbed away from the correct morphology, the collective intelligence of the agent recognizes that defect and is able to pursue it by activating various gene expressions and electrical signaling and so on, and that will progressively reduce the error, and this homeostatic loop continues to run until that error gets to be very small. And some animals, like salamanders and planaria, are extremely good at this, and they can recover from massive kinds of deformations. And so what we've been working on is treating this as an unconventional substrate for diverse intelligence, as a problem-solving system that can pursue this very large-scale goal that doesn't belong to any of the individual cells. And we've been working on the questions of where and how are these goals represented in the system, and whether or not it's possible to manipulate the system by rewriting those goals, whether it be by explicitly rewriting them, like a kind of memory inception, or to simply retrain the system by experiences. But either way, to really, rather than focus down here on rewiring the hardware, which is where most of modern molecular medicine is focused, actually to target the intelligence of the system and to look for and try to manipulate the pattern memory, or the set point that it has that allows it to do this, and then use the exact same genetically unmodified cells to show that this information is maintained by the system as an explicit description of its goals. To shift to the next piece that I wanted to show you, which is creating novel synthetic living organisms. So this is the work that our collaborators, Josh Bongard and his group at the University of Vermont, and our lab, we've started this Institute for Computationally Designed Organisms, and this is just one of the examples done by a staff scientist in our group, Doug Blackiston. And so what we've done is we've basically he's taken off some skin cells from a frog embryo, dissociated them, let them reassociate together here, and basically give them a chance to reimagine their multicellularity. And we simply ask, once again, when you have these cells in a new environment, they're skin cells, they're not genetically modified, what would they do? And you can imagine there are many things they could do. They can remain by themselves as individual cells and crawl away. They could die. They could form a monolayer of skin on the surface of the dish. In fact, they do something completely different. And what they do is they form something called xenobots. So here you can see one moving through a field of particles. This is all underwater. They're covered in cilia, so they can swim if they orient the cilia in a particular way to balance out the motion. And you can see that some of them go in circles, some of them can patrol back and forth like this. Here is some tracking data showing a number of them sort of interacting together in a dish, and you can see all kinds of different behaviors happening here. Here's an example of one in a maze. There is no water flow here. The water is completely still, so the bot goes in this direction. Here something internal happens. It turns around and goes back where it came from. So once again, there are no gradients. This is just plain water. It traverses the maze going forward. It keeps a certain distance from the wall. It takes the corner before bumping into the opposite wall. And then here, some sort of internal dynamic causes it to turn around and go back where it came from. So these are self-motile, completely autonomous little creatures made of skin that have the ability to navigate their environment in different ways. They also have regenerative ability. So here's one, and if you chop it in half almost, then here through the hinge it exerts really, really profound forces to be able to collapse it back into the shape that it had before the amputation. Of course, it regenerates back to the xenobot shape, not to the original tadpole shape. And if you look at calcium signaling, you can see that these skin cells have very active electrical signaling, very brain-like, and there are all sorts of techniques now that we can deploy to see if these two are really talking to each other. And the interesting thing about these xenobots is that if one didn't know what they were and saw the behavior and wanted to sequence the genome, one would recover only the completely 100% normal frog genome, right? So you can see here the incredible plasticity of cells as they can get together despite their normal genome, which reliably otherwise forms frogs and tadpoles, to make a new kind of creature. And one of the interesting things about these xenobots is that they don't have a straightforward evolutionary backstory. So for most animals, when we ask why do they have certain capacities, why certain anatomies, certain behaviors, intellectual capacities, the answer is always the same, because, well, for millions of years they were selected, their ancestors were selected for specific properties. And here we have a case where these capacities emerged de novo in 48 hours. Because while the individual cells certainly evolved in the biosphere on Earth, the cells had been rewarded for sitting quietly on the outside of a frog or a tadpole and keeping out the pathogens. They were not specifically selected. There were no selection pressures for being able to get together and form this kind of creature that swims around on its own and looks absolutely nothing like a tadpole or a frog. And so it's really a remarkable case of emergent behavior, emergent morphogenesis, and it gives us the opportunity in the diverse intelligence community to use these and lots of other synthetic living organisms to ask where the goals of such agents come from and where the intelligence capacities, whatever they may be. We still don't know to what extent these can be trained and so on. We don't know how much intelligence they actually have, but it provides a really interesting new model system that's free from a lot of the frozen accidents and specific selection pressures to give us something somewhat alien to practice our diverse intelligence frameworks on. And so just to get to the action plan here, what we proposed was to form a kind of process which we call a teleonomic intelligence mapping engine, that it's going to be a continuous cycle between conceptual theory, specific computational modeling, and then testing by experiment that then refines the theory and drives the production of new computation and so on, with the idea being that we will try to make a very explicit set of theory, computation, and data for applying our goal-directed type of framework to three specific, very diverse intelligences. So one is gene regulatory networks, and we and others have published analyses showing that actually gene regulatory networks should have learning capacity. So they should have a kind of simple intelligence. So one would be these regulatory networks. So this is problem solving in transcriptional space. Then, of course, the regenerating planarian flatworms, and really developing models of a collective intelligence using its pattern memory to achieve appropriate goals in morphospace. And then these xenobots, synthetic living organisms, to try to understand where their goals, morphology, and degree of intelligence comes from, given that they're created de novo in a very short time frame. And so all of this will have parallel tracks of conceptual work and theory, on which we hope to engage with philosophers of biology and lots of other people in machine learning and cognitive science and so on. And then computational modeling, so specific predictive quantitative theory here, and then the empirical work to actually test all of this with a very specific focus on being more predictive and having better control than past theories focused entirely on the molecular hardware and really not paying a lot of attention to the basal cognition in these systems to be able to do better than those existing theories. So all of the details are shown in these papers, which I will be happy to send anybody who's interested. These are our reviews discussing, in particular, the theory that goes into this approach that we hope to implement. Here are the various folks from my lab that I would like to thank, including past funders and our various collaborators on this. And we seek additional collaborators in the diverse intelligence community on several potential points. And so one thing we would really like to do is to help people apply their own framework to our various kinds of novel organisms, whether those be these kinds of two-headed planaria, which you can see here, or the xenobots, or various other synthetic organisms. We make all kinds of chimeric and synthetic constructs in our lab, and we would love to work with people to apply their own diverse intelligence framework to these organisms and see what the predictions are. And of course, we would also be happy to work with people to help design or even implement new paradigms, new learning paradigms for our novel agents. So can we train xenobots? How much memory do they have? How much predictive power do they have? What are their preferences? What are their intellectual capacities in various training assays and paradigms? We would like to work with people on better clarifying our model of goal directedness, both empirically and philosophically. There's a lot of things to still work on there, and actually work to make the cognitive light cone more quantitative. So we would like to apply this model to other people's systems and really start to build a diverse intelligence map using this kind of eigenspace in effect. So thank you so much for listening, and I look forward to discussing with you during our session.